Hello, good afternoon, everyone. Is it visible?
Yes, ma'am.
Now
It is
it should be
visible, ma'am.
visible, madam.
Yeah, okay. Thank you very much for giving me this opportunity. I am Dr. Kalaiselvi, Professor of Microbiology, as introduced by by one of your scientists. So I am been working on plant microbiome or plant-related microorganisms for past 25 years. So whatever I have an experience with this, so many years of my experience with the plant microbe interaction, I would like to share it over here. So we know like everyone is aware of the terminology microbiome now. Similar to human microbiome, the plant parts, every part of a plant system is associated with microorganisms. So we are very well aware of the human microbiome. The role of human microbiome, every part of our unit organelle is associated with normal microflora. So if there is any change in the flora, composition of the flora, there is some ailment or something, and even it is responsible for, if we have some microorganisms, very undesirable microorganisms, they are even responsible for causing cancer. So similarly, people started working the same time. People started working if there is any microorganism that is associated with whole part of the plant system, and they found out that each and every cell is associated with the microorganism, and then the number of genomes, if we take compared with plant genome, it is many folds higher in plant system. So this you may be aware of it, and the organism, the plant is having it harbors many microorganisms on the surface of the plant system as well as inside. So the one present outside is termed as epiphytes, and the one present in the inside the plant system is called as endophyte. So unlike pathogenic microorganisms, as per reports, most of the endophytes, the microorganisms residing inside the plant system, so they are mostly beneficial or commensal, and they are not pathogenic. So that's why we started working on it. In what way they are beneficial to the microorganism. So keeping this in view, we have conducted many studies. So these things, maybe you people may be aware of this microorganisms associated with below ground and above ground level. Below ground, that in the soil, the organisms nearing the soil, nearing the root system or getting influenced by the root system is termed as getting influenced by the root system is called as rhizosphere, and the soil or microorganisms adhering to the surface, on the surface, present on the surface of the plant root system are termed as rhizoplane. Okay, and the one present inside the plant system are termed as endophytes. One should not get confused with the terminology of rhizobium and WAM. Those organisms are also termed as endophytes, but then they are localized. Now the endophyte means the one present anywhere, any part of the plant system. If we introduce over the plant, that is, if we go for foliar spray, this organism will get into the plant system through stomatal opening. It will pass through the plant, get into vascular system, and then into the root system, and it may get out of the plant through root exudates. That is the definition of endophyte now. So earlier endophytes were the rhizobium or WAM, the one responsible for causing nodulation or mycorrhizal relationship. Those plants were those organisms were termed as endophytes. Now the endophyte is one capable of, it is systemic, and if we inoculate the seed, it will grow along with multiply along with the cells of the plant system, and it will get distributed throughout the plant system, right from your pollen, flower, everything. Whatever all the parts are associated with microorganisms. Ultimately, there are reports now, even we are studying about that. There are reports that certain microorganisms are transmitted from one generation to another. So that kind of findings are reported now. So this is regarding a microorganism present in the rhizosphere, and the one present outside the plant system, epiphyte, and inside are endophytes, and capable of moving from up and down. So this everyone is aware of it. With this, this work was done during early 2000 itself. Before everyone started working on microbiome, we started with this rhizosphere soil. So it was the hypothesis that shola tree species, you may be aware of shola trees, the one native to the ecosystem of Nilgiris or temperate evergreen forest ecosystem. Those tree species are ecologically and environmentally important tree species. Those have been replaced by exotic tree species like eucalyptus, and then pines, many pines, and then like battle and all those things during British colonization. And we have many plantations now. These were used for paper making, eucalyptus, paper pulp industry, and then oil industry. Then pine is also for firewood, and then other thing, battles for tannic acid, tannery, for tannery they have used. So there were large scale plantation during early, maybe after British colonization, and they are maintained after 1985, that forest policy, 88 or 85 forest policy. There were people, we cannot cut anything, so they were maintained. Because of the introduction of these species, we have lost the native species. In order to establish or restore those ecosystems, solar ecosystem, people were trying to put or trying to plant things. They have gone for managed ecosystem. They gone for planting these things in between eucalyptus and other plantations. But then they could not, the seed germination of these tree species were very poor. And if we collect and start preparing nursery also, we didn't get any seedlings. Only thing is we have to add the soil from the native ecosystem, then only it will germinate. Otherwise it will not germinate, and it will not give up the seedling at all. So, and because of that, they have introduced those nursery for in forest department. They have introduced all kinds of inputs like nutrients, enzymes, and everything. Till even then they couldn't establish a better plantation, and seed germination was very poor. And only upon addition of soil from shola ecosystem for preparing nursery, as well as for nursery bed, as well as for polybags, then only they could establish the nursery. And from that we found out there was another observation. Wherever we had mugreen or algal mat, there was germination of these seedlings. So we thought some factor of microbial origin is responsible for facilitating or eliciting germination of these recalcitrant tree crops. So with that idea, we have started. We have isolated microorganisms, rhizosphere microorganisms, from very important tree species like Nothopodia. It is an anti-cancerous property having plant species. Another two important ecological species like Eleocarpus oblongus and Evadia luni intenta. All those things. And we just wanted to find out whether the rhizosphere microorganisms associated with these three species at different altitudes of Nilgiris or one and the same, or differs. So with that we have done. So this is pictorial representation of the microbial population in different shola ecosystem only. The type of ecosystem is one and the same, that is shola of different ecosystem, longwood, taiga, and thiosola. So longwood is located in Kotagiri, taigasola is in the location I forgot, Manjur. Thiosola in Manjur and Taigasola in Kunnur. So with different altitudes, we just wanted to find out whether the organism associated with rhizosphere are one and the same, or they vary with the soil type as well. So this is the bacterial flora we got with Eleocarpus oblongus. This is from rhizosphere soil of Eleocarpus oblongus, and this is from nearby bulk soil or non-rhizosphere soil. You can see the difference. And from nearby sholas only, we got the samples from grasslands. And blue gum is nothing but eucalyptus, and black wattle plantation nearby only from Kalaiselvi. So you can see the entirely different kinds of microorganisms, bacterial flora. This was in 2002. So this is in Tiger Woods Solar. We got entirely different kinds of bacterial flora with more of pigment producing microorganisms. So this similarly in case of fungi. So this is in this particular these isolates were obtained from grassland of nearby Shola ecosystem. This is from Shola. So Shola soils were dominated with the penicillium, and grasslands had more of fusarium. That was the qualitative difference we could observe in those days. And this is from another Thai Shola ecosystem. From the same rhizosphere, we got a different kind of fungal flora. So this is from tea plantation, nearby tea plantation, Shola ecosystem, nearby tea plantation. So this is from blue gum and black wattle plantation. So entirely different types of fungi we could get it. So this is how the kind of plant we are going to have a pronounced effect on fungal and bacterial flora. That's how we found out this. And we have also developed cultures for eliciting germination of these trees seeds under nursery condition. So this is regarding one kind of observation we have got with the Shola tree species. And another important thing is actually we have worked with forestry species as well as agriculture crops. I'm just talking about forestry species first, and then I'll tell you about the agriculture crops wherein we have found out certain plant microbial, specific peculiar microbial interactions. So another important important tree species is Melia dubia. It is Malay bamboo. You might have heard of it. Some of you may be aware of it, which is used as a alternate for cashew nut. There's a pulpwood species. It's a fast-growing organism, fast-growing tree species again. Here again, seed germination is a problem. Seeds will germinate only under the tree species. They will not germinate even if you go for any treatment like breaking dormancy by hot water, acid treatment, cow dung application, and all those things, all those technologies they have tried, still they couldn't. All these technologies were found to be futile. So because of that, again, we got an idea that microorganisms may induce or favor or facilitate germination over here. And depending on that, we have collected seeds from natural ecosystem, parts rather from natural ecosystem, and we got the actually see this is the part. It has a hard seed coat, and imbibition is a problematic thing. Still, even after imbibition, there was no germination. So inside we have 3 or 4 locules. From that we get seeds like this. So either we have to break the hard seed coat and get this. During this, we may lose the viability of the seeds also. So in order to enhance the germination capacity of this particular species, we have done certain experiments. We got the fruits, ripened fruits from natural ecosystem. We have subjected those fruits to microwave treatment, microwave boiling for this is for 2.5 minutes, 5 minutes, 10 minutes, 15 minutes, and then 20 minutes. And there was no, 2.5, 5, 7.5, 10, 15, 20, and 0. It is no treatment of microwave and no treatment of microorganisms. So first we have subjected this fruits to microwave treatment by boiling in microwave treatment for 2.5, 5 minutes, 7.5, 10, 15 minutes, and this is 20 minutes. There is no microwave treatment. This is a control, absolute control treatment. So we have also isolated microorganisms from the seed pod by enrichment culture technique in order to decompose the seed material so that it will facilitate imbibition and the germination of the seeds. So we got the microorganisms, cellulose and lignin degrading microorganism. After this microwave treatment, we have pelletized the foods with this and gone for seed germination studies. So with 2.5 minutes, there was no germination. And this is the number of days taken for germination and all those things. So in case of 5 minutes, it took around 45 days to germinate, and ultimately we got 52% germination. And in case of 7.5 minutes, within 10 days, I'm sorry, within 10 days, we got the germination. This is for completion of the germination. So within 10 days, we got the germination percent of 68%, and later on it got reduced. And this is maybe the seeds are getting affected by microwave treatment. And within 10 minutes, 2.5 minutes, within 12 days of incubation, we got germination of this, and it took 45 days to complete. Here 68% germination within 35 days. So that's how we have developed a technology for improving germination or facilitating or triggering germination in Melia dubia with microorganisms. Prior to that, we have gone for microwave treatment. So these are the photographs of different treatments. So there is yet another technique on forestry. Normally we go for growing the organisms through clonal multiplication. In order to get clones, you may be aware of, in order to get homogeneous population, we go for cloning. But for inducing rooting of these clones, we normally go for chemical inputs like IAA and IBA. In order to reduce that, or in order to eliminate that, we thought we'll develop some microorganism that will elicit rooting. So we have gone with vermicompost and mycorrhizal consortia for Casuarina. So we got the these consortia, vermicompost and mycorrhizal consortia, could elicit rooting very well in these things. So this is another observation. So we have also studied the mycorrhizal status of certain introduced tree species. Cymaruba claca, it is normally called, in Tamil it is called as Surgamaram. Seeds are rich in oil, around 60% oil. It has got oil. This is it is edible, but only thing is some inhibited substances are there that those things are to be removed. Otherwise, it can be a very good source for biodiesel production. So we just wanted to find out the mycorrhizal status of the natural stand. So there was no infection in the natural stand. The roots had no mycorrhizal infection. Suppose we have inoculated, and we could find 86.5% infection. This is the infection with lot of vesicles and vesicles in it. That is the dependency of, that is the response of the plant to mycorrhizal infection. So that's how we found out that even mycorrhiza, this plant could respond to mycorrhiza and improve. So in case of Jadroba also, it was in 2005-6, I think, I remember. So here also we have instead of seeds, they have gone for clones in order to get homogeneous population, or clones from higher yielding natural stands. So in order to improve the rooting, we have used Azospellum and BAM. This is uninoculated, and this is inoculated. Mycorrhiza and Azospellum bacteria inoculated cuttings. So even rooting was very greater because of these inoculants, BAM, Azospellum, bacteria. That is the impact of the microorganisms associated with plants. So far it was impact of microorganisms in improving growth of cuttings, or improving rooting of sprigs, or improving germination of recalcitrant seeds. So microorganisms, you may be aware, are normally used for improving plant growth through nutritional modes. Maybe they are nitrogen fixers like azospellum, bacter, and rhizobium. Rhizobium, which is recommended for leguminous crops, and phosphobacteria, which enhances the availability of phosphorus in soil, and mycorrhiza that even that is involved in mobilizing phosphorus. So microorganisms as biofertilizers are recommended basically for improving nutrition. Besides, microorganisms are also used for improving seed germination that you have seen now, and for improving rooting of cuttings or sprigs that also you have evidenced now. So besides, uh, after I think in the beginning of 2013 or 14 also, the work on plant microbial interactions enhanced, it was given importance and thrust, and people started working on many aspects of microorganisms. As we have discussed earlier, every part of the plant cell is associated with microorganism, and mostly they found out that they are not pathogenic. They are not pathogenic microorganisms, mostly commensal in organisms. So because of that, we just wanted to find out what is the role of these organisms inside the plant. So how do they move from up and down? Once it is given as foliar spray, how this organism get into root system or maybe into rhizosphere soil. So what is the role of these organisms? In order to elucidate all those things, we have conducted certain experiments. One of the studies in which we just thought that maybe the apoplast, that is the fluid present in between apoplast, the region present in the cell wall and cell membrane, in which apoplastic and symplastic movement, all those things will occur. Nutrients will be transferred from one cell to another, and they may have an interaction with the vascular system through which things will move from up and down. Root system, that is the connecting point of shoot system and root system. So we thought these besides metabolites and nutrients, these apoplasts might harbor microorganisms or beneficial organisms, and they may contribute to the beneficial, or they may contribute to the if they may improve the plant growth. So with that, the idea, we have started working on three different kinds of rice varieties. One is moisture stress susceptible or normal variety. It is rice 451. Another two varieties of Anna R4 and IR 64. These two varieties are resistant to drought. Anna R4 rice variety and IR DRT64 is also resistant to drought. And while 451 was 415 rice variety is susceptible, or it is a normal variety. It cannot tolerate the moisture stress or drought tolerance. So just we wanted to isolate microorganisms, apoplastic microorganisms from these crop species, and wanted to develop, is there any first find out whether is there any difference in microbial load of the apoplast, and if whether if we introduce the microorganism from this into the susceptible one, it will elicit moisture stress tolerance. So in order to develop moisture stress tolerating or imparting microorganism, we have conducted this experiment, and totally we have got. So you can see the microorganism, an NAC, in the sense the organism was isolated from apoplast of rice using Na medium, LB medium, TSA medium, R2A medium, LB drop, similarly TSA and all those things. So this is from Co51. C is for Co51, R2A, A is for Anna R4, and finally, I'm sorry. So another one is from DRT. So we have totally got 41 species, 41 bacterial isolates using different media with different concentration in order to isolate both the oligotrophic and other kind of microorganism, proteotrophic microorganism, bacteria from apoplast of rice. So of 41 isolates, Around 26 were found to be Pseudomonas aeruginosa. It is an opportunistic pathogen. So we have removed all those things. We have concentrated on rest of the 26 bacteria. We have assessed the beneficial characteristics like nitrogen fixation, phosphate solubilization, potassium release, sulfur oxidation, both nutritional and non-nutritional modes of beneficial diets of the organism, cider production, hydrogen cyanide production, and their antagonistic activity against important rice pathogens, and all those things we have studied. And ultimately, we reduce this. We have screened 12 organisms. Out of 12, Spingobium, the one from R2A, and one Spingobium with an R4, another with the IR DRT64 we have got. So we have concentrated on an organism or bacterium, Spingobium from IR DRT64. So we have gone for further studies with this. So this is the thing, IR64DRT, we have got an organism called a Spingobium enochiae. Through molecular work, we have found out the organism as Spingobium enochiae. And it is capable of, these are the beneficial characteristics of this. It could tolerate up to minus 5.5 megapascal, which was created using PEG 6000. And it is capable of growing in different wide range of pH 2 to 10. And temperature of 4 to 55 degrees centigrade, and it's capable of tolerating salinity up to 1 molar and chromium of 10 ppm. And it is capable of producing growth hormones like IAA, GA3, and abscisic acid, and the enzyme called ACCD, which is responsible for imparting drought resistance. So this is one thing. So when this organism is grown under normal condition, these are the characteristics. Of course, this particular organism was not a nitrogen fixer, phosphate solubilizer, or potassium releaser. It is not involved in those nutritional characteristics. It was never negative for all those properties. So when it was grown under stress, moisture stress condition of minus 5.5 megapascal, there was a 5-fold increase in gibberellic acid production. That was the peculiar characteristics of the bacterium. And produced a lot of polysaccharide, which is responsible for, an important property of this organism to tolerate moisture or any stress. And under stress, it was capable of accumulating compatible solutes like proline, glycine, betaine, and other things. So those things we have found out. So these are some of the beneficial things we have characterized for this organism. So we just wanted to find out these are the characteristics of this, and its ability to tolerate moisture stress of this minus 5.5 megapascal. Whether this organism will impart drought tolerance to the plant, that we wanted to study. So we have used this cell pellet of this. We have grown this in liquid media, got the cell pellet, and used for pelletizing O51. It is moisture stress thing, 451, and we have inoculated that. This is inoculated and uninoculated. Spingobium, you may be aware of, it is yellow pigmented. When we have gone for this, within 20 days, we got a plant with yellow pigmented roots. So it colonized the root. So we just wanted to find out whether it is colonizing on the root system alone, or even it is present in the shoot system. It is just to verify the endophytic nature of it. So we have analyzed the colonizing pattern in leaf and root system, stem, and then root system also. So in case of leaf surrounding the stomata, we could see the colonizing potential. It was analyzed through SIM. And in vascular system also, this is uninoculated control. You can see the clear vascular system here. And here, this organism got filled up. The vascular system got colonized fully by this organism. And not only that, even intercellular parenchymatic cells, in which we have found a lot of organisms which are missing here. This is the control thing. So this is how we have affirmed that there is a colonization in root system as well as shoot system. So then we found out that it is endophytic, that we have confirmed it. And later we just wanted to find out its potential to impart a drought resistance or moisture resistance to the crop species. So the same co-51 was inoculated with three different kinds of microorganisms. One is T1 is control with the flooded condition, and T2 is one inoculated with another moisture stress mitigating bacterium. This is PPFM. And this is Microbiome, the one what we have isolated. And another one is Bacillus aeugitoninus. It is also involved in drought mitigation, drought mitigation. And then the last one, T5, is moisture stress grown under uninoculated condition. It is grown, this particular plant species, which was grown under drought stress without any inoculation. These three are grown under moisture stress with three different microorganisms. This particular one is grown under flooded conditions. So this is one particular control, and T5 is also, it is negative control, it is positive control. So this is co-51 grown under flooded condition without any inoculation. These are grown under moisture stress condition after 70 days of 70 days of sowing. Moisture stress was given 75% of the field capacity was the moisture stress given. The level of stress given was 75% of field capacity. So in that time, how these organisms and the behave. So this is normal. It has reached the harvesting stage or maturity stage. This is the root system. And this is the one inoculated with the methylarubrum, and this is the one with the PPFM. So we could see the root volume is similar to that of one grown under, almost similar to that of one grown under flooded condition. And the morphological characteristics, if we see, they are upright, and the pigment content, chlorophyll content was also very high, and it was green even after 130 days of stress. So that was the thing. So you can see the lesser root volume in this bacillus here also. This is uninoculated control grown under stress. So these are the observations we have got, and we have confirmed this with the gene expression studies also. So this is grown under control condition without any inoculation. This is with inoculation. So expression of moisture stress related genes in S Yanakuye inoculated under normal and moisture stress condition. Okay, this is under a normal condition, flooded condition, and this is with moisture stress condition. So we have seen the higher expression of GST, glutathione transferase, which is responsible for reduction of stress or antioxidants produced during stress. So that's how we found out that this organism is capable of modulating the physiology during stress condition, and it imparts this because it is capable of producing IA and GA in a larger quantity during stress. It might have contributed to higher root volume as well. So this was our sorry, this was our observation with this particular work. So this work is still going on. We just wanted to confirm it under laboratory condition. So there is another thing with another terminology you might have heard about now. This microbiome, and then epiphyte, endophytic organism, endophytes. There is a small, there is another terminology called core microbiome now. People say about, talk about core microbiome. It is nothing but, once again, please. Core microbiome is nothing but one that is closely associated with the particular genotype. The expression of it is not affected by, it is expressed or it colonizes the plant irrespective of the ecosystem or soil type in which it is grown. Suppose we find out the core microbiome. If we introduce that microbiome, it will take care of the growth of the bacteria, that is the growth of the plant. That is the idea behind it, because this is considered as the core microbiome. It is considered as a hub microbiome. which may not be involved directly in improving plant growth by nutritional or non-nutritional modes, but however, it is responsible for recruiting beneficial microorganisms. Suppose we introduce this, we need not apply any biofertilizers. It will recruit the microorganisms from the soil itself. That is one thing. Otherwise, if a particular microorganism is not present, beneficial organism, if we introduce as biofertilizers also, these organisms will help in recruiting those things. So with this hypothesis, these two concepts in mind, we have worked on this. We are still working on it also. So the popular maize variety CoH6 we have taken, and the parental forms of male and female also we took, and we analyzed the total microbiome of it using NGS, that is Illumina sequencing. 16S Illumina sequencing technology, we have sequenced and found out the bacteria and actinobacteria associated with the seeds of maize CoH6, and found out 1 on 6 of total 1 and 6 or bacterial flora were found to be common to all, that were present in all the things. So those are termed as core microbiome. So we just wanted to find out, this is based on metagenomic approach. So we just wanted to find out, for practical application, we need to have a culturable microbiome. This is based on the genomic and using bioinformatic tools. Sorry, we found out the organisms or core microbiome, the level of core microbiome present in. This is 1 on 6 we have found out. So because since we wanted to have a culturable thing so that we can introduce those microbes as inoculants for improving core microbiome level of hybrid, we need to introduce this for male as well as female. We have to inoculate male and female so that it will come to hybrid. So we have gone for cultural microbes also. We have collected culturable microorganisms from bacteria from these 3 sources, male, female, and hybrid seeds. Those 12 were identified as these things: Microbacterium, Bacillus subtilis, Teculins villagensis, Enterobacter rohangampi, Enterobacter cloacae, and likewise we got Pristia, Bacillus, 12 organisms as very distinct and culturable things, culturable microorganisms from it. By comparing the culturable microbiome with core microbiome of metagenomic approach, we have found out the culturable core microbiome. We have got totally 12 endophytes. Out of 12, we have found out 7 as core microbiome. By comparing metagenomic approach with culturable microbiome. So those microorganisms are, we got Bacillus, then Bacillus, all the bacilli, 5 species of bacilli were found to be so out of 12, 5 species were found to be Bacillus, and 1 alkaligene species that was also found to be core microbiome. So out of 12, 7 were found to be core microbiome. So we wanted to just introduce these 7 species for both male and female and find out whether these organisms are present in hybrid under laboratory conditions by tagging the organism and monitoring the presence of different generations. First, we have to introduce this for male and female, and we have to find out in hybrid. So this is how we just wanted to find out the presence of core microbiome. So that is one side. Other side, it is not a new thing. We recommend Rhizobium vam phosphobacteria mycorrhiza for plants. So during this process, Rhizobium, we know it is responsible for enhancing nitrogen fixation or nitrogen mutation of the plant by improving nodulation. Other side, mycorrhiza improves in phosphorus nutrition and other immobile nutrients also, mobilize immobile elements and improve nutrition. Besides, It's now it's not now, maybe 15-20 years now onwards, people started like a decade also, people started working on induced systemic resistance or SCR, systemic acquired resistance imparted by these organisms. So they not only improve the plant system through nutritional modes, they also impart resistance against insect attack and pathogen. Of course, we know there are plenty of reports, and insect attack. So we just wanted to find out whether these organisms have whether they have any effect on improving tolerance limit of the plant to Spodoptera litera infection in black gram. So we have used rhizobium specific to black gram, as well as Glomus interradius, AM species, and we have confirmed the presence of VAM after confirming the presence of nodule and VAM. We have introduced the Spodoptera litera, and we have studied the metabolites profile of infected tree species, infected leaves as well as uninfected leaves, because there is systemic induction of resistance in tolerance in other nearby unaffected leaves also. So in order to find out that, we have studied the metabolic response, metabolic physiology of the nearby nearby leaves as well as affected leaves, affected plants and nearby plants also. So this is how we have formed the induction of systemic resistance in this. Some disturbance. Hello, there is some disturbance. Right. Whenever plants are exposed to any stress, maybe biotic or abiotic stress, reactive ozone species will be generated. That is responsible, like hydrogen peroxide singlet for damaging the thing, further damaging the thing. So we just wanted to confirm the production of all those things. In this study, we have done this. So maybe here we have analyzed the same thing. I'll just close this and share with the So one of the mechanisms by which the introduction of rhizobium and fungi protect plants from insect damage is by enhancing the by improving the morphology of leaf, number of uh, trichomes. They will increase the trichome density, thereby they'll give protection against movement of larvae. So we just wanted to find out that. We have studied this. This is in control without Spodoptera and with Spodoptera attack. And in case of rhizobium, it is systemic thing we have analyzed. It is not the one affected by, the leaves are not affected by Spodoptera. It is a systemic study we have done. This is with rhizobium. When we have gone for rhizobium plus Spodoptera, there is increase in trichome number. With glomus, further an accelerated thing when we have introduced glomus and rhizobium together. The number of trichomes increased. The trichome number increased